Hello again and welcome back. Today we're going to take up Chapter 24 on lipid biosynthesis. The essential question for this chapter is what are the pathways of lipid synthesis in biological systems? The outline of the chapter looks like this. We'll first discuss the synthesis of fatty acids and then ask how complex lipids are synthesized. We'll also talk about eicosanoids, 20 carbon molecules that induce pain in organisms. And then we'll talk about cholesterol synthesis and lipid transport throughout the body. And we'll finish up by talking about bile acids and steroid hormones. How are fatty acids synthesized? Well, it turns out that fatty acid biosynthesis and degradation pathways are different. As in the cases of glycolysis and gluconeogenesis and glycogen synthesis and breakdown, fatty acid synthesis and degradation have to go by different routes so that each of the pathways will be favorable with a large negative delta G. There are four major differences between fatty acid breakdown and fatty acid synthesis. First of all, intermediates in synthesis are linked to SH groups of acyl carrier proteins, which we haven't met yet. But this is actually quite similar to the chemistry of coenzyme A, because it's a thiol group and an acyl carrier protein replaces the thiol group of CoA. The thiols of acyl carrier proteins replace the thiols of coenzyme A in fatty acid oxidation. Synthesis of fatty acids occurs in the cytosol, whereas, as we discovered in Chapter 23, fatty acid breakdown occurs in mitochondria. The third difference is that enzymes of synthesis are one polypeptide, the so-called fatty acid synthase. The fourth point is that biosynthesis uses NADPH, NADP+, whereas fatty acid breakdown uses NADH and NAD+. The design strategy for fatty acid synthesis is as follows. First of all, fatty acid chains are constructed by the addition of two carbon units derived from acetyl-CoA. These acetate units are activated by carboxylation with CO2 to form malonyl-CoA at the expense of ATP. Remember, carboxylations are energetically expensive, so ATP is required, and we'll see that malonyl-CoA is the committed metabolite in fatty acid synthesis. The addition of two carbon units to the growing chain is driven by decarboxylation of malonyl-CoA, and the elongation reactions are repeated until the growing chain reaches 16 carbons in length, which of course is a palmitic acid. Other enzymes then add double bonds and additional carbon units to the chain. This figure will appear to be daunting at first, but it's really easy to work your way through it, because you've seen much of this before already in metabolism. Let's say at the beginning that the blue arrows indicate carbon substrate flow, and the red arrows indicate electron flow. And it's important to realize that for fatty acid synthesis, you need both carbon units and electrons. Biosynthesis is reductive and anabolic, and it requires electrons, as we've said before. So start at the bottom and look at glucose and realize that in glycolysis, a few electrons are produced to produce an NADH. Those are converted by malate dehydrogenase into malate, and those electrons flow through the malic enzyme to NADPH, which is the substrate that provides electrons for fatty acid synthesis. What about the carbon units? Start at glucose again and realize that the pyruvate produced in glycolysis can enter the mitochondria and then, as we've seen before, go through the pyruvate dehydrogenase reaction and the TCA cycle, and that citrate can come out of the TCA cycle, cross the intermitochondrial membrane, be decomposed by ATP citrate lyase, and thus provide acetyl-CoA units for the synthesis of fatty acids. In addition, fatty acids themselves can produce fatty acyl-CoA and then fatty acyl-carnitine, which crosses the mitochondrial membrane, as we've seen, and goes through fatty acid oxidation to produce acetyl-CoA. That acetyl-CoA can enter the TCA cycle, but some of that citrate can be exported too and provide acetate units for fatty acid synthesis. So this diagram is not as complicated as we might have thought. It's pretty easy to see where the electrons are coming from and where the carbon substrates are coming from. This is the citrate-malate pyruvate shuttle, and it's the essence of how carbon units and electrons are provided for fatty acid synthesis. Carboxylation of acetyl-CoA to form malonyl-CoA is catalyzed by the enzyme known as the acetyl-CoA carboxylase. This is the ACC enzyme. Acetyl-CoA carboxylase abbreviates to ACC. And this enzyme commits acetate units to fatty acid synthesis because it synthesizes malonyl-CoA, a committed metabolite. Carboxylation of acetyl-CoA to form malonyl-CoA is the irreversible committed step in fatty acid biosynthesis. The ACC enzyme uses bicarbonate and ATP as substrates, and that should immediately make you think of biotin. And in fact, biotin is a required coenzyme for the ACC enzyme, as we'll see. The E. coli enzyme is best understood, and it has 3 subunits. The animal enzyme is one polypeptide with all 3 functional parts, these being the biotin carboxyl carrier protein, the biotin carboxylase, and the so-called carboxytransferase. This shows you the quaternary structure of the E. coli biotin carboxyl carrier protein biotin carboxylase complex. The BC subunits are orange and blue, and the BCCP subunits are pink and yellow. This is the biotin carboxylase domain from human acetyl-CoA carboxylase 2, with the A subdomain in blue, the B subdomain in red, the AB linker in green, and the C subdomain in yellow. The carboxyltransferase domain dimer of the acetyl-CoA carboxylase 1 from Saccharomyces is shown here. The N and C subdomains of one monomer are cyan and yellow, whereas those of the other monomer are purple and green. Coenzyme A is shown as a ball and stick model in one of the subunits right here. The acetyl-CoA carboxylase reaction produces malonyl-CoA for fatty acid synthesis. And this really means that acetate units are committed to fatty acid synthesis by formation of malonyl-CoA. This is a carboxylation that uses ATP and bicarbonate, so you know that biotin is required. And here's the mechanism for the acetyl-CoA carboxylase reaction. Bicarbonate has to be activated in this reaction, and it's activated by its reaction with ATP to form carboxyl phosphate or carbonyl phosphate, which we've seen before. This is an activated form of a bicarbonate or carboxyl group. Once again, the formation of carboxyl phosphate illustrates the importance of activation of groups for transfer in biological metabolism. Biotin attacks the carbonyl carbon of the carboxyl phosphate to form N-carboxybiotin, as we've seen before. And then the N-carboxybiotin is attacked by the anion of an acetyl-CoA, and this illustrates CoA chemistry again. Remember that acetyl-CoA is acidic at its methyl carbon and can form the carbanion shown here, which then attacks the carbonyl carbon of the N-carboxybiotin to form the malonyl-CoA product. The acetyl-CoA carboxylase in E. coli, as we mentioned, consists of 3 subunits. In the ACC reaction, the biotin ring acquires carboxyl groups from carbonyl phosphate on the biotin carboxylase subunit and then swings across the gap here to the carboxyl transferase to deliver that 1 carbon unit to the acetyl-CoA to form malonyl-CoA, as shown here. Biotin is essentially a flexible tether that delivers carboxyl groups from the carboxylase to the carboxyl transferase. In animals, acetyl-CoA carboxylase, or ACC, is a long, active filamentous polymer formed from inactive protomers. Each of these protomers contains the biotin carboxyl carrier moiety, the biotin carboxylase, and carboxyl transferase. And since this is a committed step, you would expect it to be highly regulated, and it is. ACC is carefully regulated. Palmitoyl-CoA, the product of the reaction, favors monomers, and citrate favors the active polymeric form. This is pure energetics on the part of the cell. When there's enough palmitoyl-CoA, monomers are favored, and the ACC is inactive. On the other hand, when citrate is plentiful, it favors the active polymeric form, and fatty acids are produced. Phosphorylation modulates citrate activation and palmitoyl-CoA inhibition. The regulatory effects of citrate and palmitoyl-CoA are dependent on the phosphorylation state of the acetyl-CoA carboxylase. The animal enzyme is phosphorylated at 8 to 10 sites on each enzyme subunit. The unphosphorylated enzyme has a high affinity for citrate and is active at low citrate concentrations. The unphosphorylated enzyme also has a high Ki for palmitoyl-CoA, and high palmitoyl-CoA is thus required to inhibit. The phosphorylated enzyme, on the other hand, has a low affinity for citrate, and high citrate concentrations are needed to activate. The phosphorylated enzyme has a low Ki for palmitoyl-CoA and is thus inhibited at low palmitoyl-CoA concentrations. This is a schematic showing the acetyl-CoA carboxylase with the domains and phosphorylation sites indicated, along with the protein kinases responsible. Notice the number of different kinases that are required to carry out the phosphorylations that regulate this complex enzyme. The activity of acetyl-CoA carboxylase is modulated by phosphorylation. The dephosphoform of the enzyme is activated by low citrate and inhibited only by high levels of fatty acyl-CoA. In contrast, the phosphorylated enzyme is activated by high levels of citrate. Fatty acid synthesis does not use coenzyme A. Instead, it uses a protein analog of coenzyme A known as an acyl carrier protein, and acyl carrier proteins carry the intermediates in fatty acid synthesis. The acyl carrier protein was discovered by P. Roy Vagelos, who rose to become the CEO of Merck in the United States. In E. coli, the acyl carrier protein consists of 77 residues and has a phosphopantothenium group on it, which we'll see. In terms of its function, it's really just a large coenzyme A. Figure 24.6 compares the acyl carrier protein and coenzyme A itself. And here you see that both coenzyme A at the top and the acyl carrier protein on the bottom consist of a phosphopantothenium group linked to other moieties. In the case of coenzyme A, this part of the molecule is for purposes of binding to the enzymes that use it, and similarly, with the acyl carrier protein, you have a place to put the phosphopantothenium to carry out the chemistry that's required. But in both cases, the business end of the molecule is the thiol on the left here. These thiols, of course, produce thioesters. These thiols are prolific. These thiols, of course, form thioester intermediates with acetyl groups and other acyl groups. Fatty acid synthesis in mammals occurs on a homodimeric fatty acyl synthase 1, also known as FAS1. The FAS1 consists of 270 kilodalton polypeptides, which contain all the reaction centers required to produce a fatty acid. On the other hand, in yeast and fungi, the activities of fatty acyl synthase are distributed on two multifunctional polypeptide chains. In plants and bacteria, the enzymes of the fatty acyl synthase are separate and independent, and this collection of enzymes is referred to as the fatty acid synthase 2, or FAS2. The individual steps of fatty acid synthesis are similar across all organisms. The mammalian pathway shown in figure 24.7 is a cycle of elongation that involves 6 enzyme activities. Elongation is initiated by transfer of the acyl moiety of acetyl-CoA to the acyl carrier protein by the malonyl-CoA acetyl-CoA acyl carrier protein transacylase, which fortunately simplifies to the MAT for the malonyl acetyl transferase. This enzyme also transfers the malonyl group of malonyl-CoA to the acyl carrier protein, as we'll see. This figure looks daunting. It shows the pathway of palmitate synthesis from acetyl-CoA and malonyl-CoA, but we can break this figure down and make it understandable. Start at the top and see that acetyl-CoA reacts with the malonyl acyl transferase to form an acyl carrier protein adduct, an acetyl group linked to the acyl carrier protein. That 2-carbon unit is transferred to the ketoacyl synthase to produce this intermediate, and then that combines with a malonyl-CoA also linked to an acyl carrier protein, and this 2-carbon unit and this 3-carbon unit combine with release of CO2, a decarboxylation that is spontaneous, to form the acetoacetyl acyl carrier protein. This then goes through a reductase reaction that takes the carbonyl to a hydroxyl and then removes the elements of water across this bond to form a double bond as shown here. That double bond is reduced to form a single bond to form butyryl acyl carrier protein, and then the cycle repeats over and over until a palmitate is produced. Notice the similarity in the preparation of the acetyl-CoA for this reaction and that of the malonyl-CoA. The acetyl-CoA forms a thioester by the action of the malonyl acyl transferase, and so does the malonyl-CoA. And then both these intermediates react in the beta-ketoacyl synthase reaction to form the product here. On the bottom half of this pathway, we see that the reactions are very similar to what we've already seen in the TCA cycle and in fatty acid oxidation. Starting with the carbonyl group in the acetoacetyl acyl carrier protein, a reduction occurs to form a hydroxyl group, water is removed, and a double bond is turned into a single bond. This is the same set of 3 reactions that we've seen over and over again. In this case, it's going in the direction opposite to the TCA cycle. If we ran it backwards, we'd be taking a single bond to a double bond, hydrating across, and then oxidizing. In this case, it goes the other way, from a carbonyl to a hydroxyl to a double bond and to a single bond. And this is the key to understanding the similarity of fatty acid synthesis, fatty acid degradation, and the TCA cycle. This shows a mechanism for the mammalian ketoacyl synthase. A hydroxyl group on the malonyl acyl transferase attacks the carbonyl carbon of this thioester intermediate and forms this intermediate, which is attacked by the SH group of an acyl carrier protein to produce a new thioester as shown here. The ketoacyl synthase attacks that, while the malonyl acyl transferase attacks the carbonyl carbon, and the result is a new thioester formed on the ketoacyl synthase. The third step involves thioester exchange between the ketoacyl synthase and the acyl carrier protein with the acetyl group, and a linking of malonyl-CoA to the MAT as shown here. Attack by the SH group of an acyl carrier protein on the malonyl-CoA forms this intermediate, which can decarboxylate with attack on the carbonyl of the acetyl group on the ketoacyl synthase. Loss of CO2 is spontaneous, and the product, the butyryl acyl carrier protein, is formed. The fatty acid synthase in all organisms is complex, and it's worth taking a few minutes to look at some of the structural information that's available. The fungal fatty acid synthase looks like this. This block diagram shows where the individual enzyme activities are located. And this is a cartoon illustrating the structure of the mammalian fatty acid synthase. Notice there's some gaps here and here. We don't have a complete structure yet for this complex mammalian synthase, but a partial structure is available. This is a lovely structure of the fungal fatty acid synthase, and you see in the middle of the structure struts that hold the entire structure together with these pairs of helices here and here and here. And these are really coiled dimers that stabilize the structure. They're referred to here as stabilizing struts. This is what's known of the mammalian fatty acid synthase. It's a homodimer, and it's X-shaped, and the acyl carrier proteins and the acyl carrier protein domains are probably located adjacent to the KR domains shown here and here. But this structure is incomplete at this point. Now that we've made a 16-carbon fatty acid, a palmitic acid, we can ask, how is palmitic acid elongated and how are unsaturations added to the fatty acid chain? Additional elongation occurs in mitochondria and in the endoplasmic reticulum of animal cells. The introduction of cis double bonds is different in prokaryotes and eukaryotes. Prokaryotes use an oxygen-independent process, whereas eukaryotes use an oxygen-dependent process. In E. coli, double bonds are added while the site of attack is still near a functional group, namely the thioester. On the other hand, eukaryotes add double bonds to the middle of the chain, but they need the power of oxygen to do it, as we shall see. Elongation of fatty acids in mitochondria is initiated by a thiolase reaction, as shown here. The acyl-CoA adds a 2-carbon unit to form a beta-ketoacyl-CoA, which can undergo reduction to form a hydroxyacyl-CoA, and then a double bond formation occurs with removal of water, and the double bond is reduced to a single bond, as we've seen already. In prokaryotes, double bonds are introduced into the growing fatty acid chain by specific dehydrases. Palmitoyl acyl carrier protein is synthesized by a sequence of reactions involving 4 rounds of chain elongation, followed by double bond insertion by a beta-hydroxydecanoyl thioester dehydrase, and 3 additional elongation steps. Another elongation cycle produces cis-vaccinic acid as a cis-vaccinoyl acyl carrier protein, as shown at the bottom. Unsaturation reactions occur in eukaryotes in the middle of an aliphatic chain. And you may find this diagram to be puzzling at first, but we're going to walk through it carefully. What's the point of the reaction? We're trying to introduce unsaturation in a saturated chain, so we're taking a single bond to a double bond, which is an oxidation. So the real purpose of all this is an oxidation, but you start over on the left and find out that electrons are involved in this process, as if it were a reduction. What's going on? The secret lies over here on the right, when you see that oxygen molecules are reduced to a pair of water molecules, and one oxygen molecule converted to 2 waters is a 4-electron reduction. The conversion of a single bond to a double bond here only provides 2 electrons for this process, so the other 2 electrons are provided by an NADH. Removal of electrons from NADH produces NAD+. Those electrons are transferred to a cytochrome B5 reductase, and then to a cytochrome B5, and then to a desaturase enzyme, and finally to the oxygen in the reduction that's shown here. So interestingly, an oxidation to form a double bond involves reduction as well. The formation of additional double bonds in eukaryotes looks like this. Arachidonic acid is synthesized from linoleic acid. In the first step, linoleic acid is converted to a linoleoyl coenzyme A by an acyl-CoA synthetase, which consumes an ATP and produces an AMP and pyrophosphate. And of course, the pyrophosphate hydrolysis drives this reaction forward. So the CoA ester is formed, and then desaturation occurs, removal of a pair of electrons to form a double bond, producing a linolenoyl CoA with the double bonds at the 6, 9, and 12 positions. Elongation occurs next, with a malonyl CoA donating essentially a 2-carbon unit with decarboxylation to produce this structure that's 2 carbons longer than the previous one, and that moves the double bonds to the 8, 11, and 14 positions. Then a second desaturation occurs to introduce a new double bond at the 5, 6 position. Hydrolysis of the CoA ester produces the final product, arachidonic acid. And this sequence of reactions explains why the double bonds in arachidonic acid are at the 5, 8, 11, and 14 positions, whereas all of the other typical fatty acids with unsaturations have double bonds at the 9, or the 9, 12, or the 9, 12, and 15 positions. Arachidonic acid is different. If you're interested at all in healthy eating, you've certainly encountered omega-3 and omega-6 fatty acids in your reading. These are essential fatty acids with many functions. Linoleic acid and linolenic acid are termed essential fatty acids because animals cannot synthesize them and have to acquire them in their diet. Linoleic acid is the precursor of arachidonic acid, and both of these are termed omega-6 fatty acids for a reason that we'll see in just a minute. Linolenic acid is the precursor of eicosapentaenoic acid, or EPA, and docosahexaenoic acid, or DHA, and both of these are termed omega-3 fatty acids. Omega-3 fatty acids are cardioprotective, anti-inflammatory, and anti-carcinogenic. Omega-6 fatty acids are precursors of the prostaglandins and the thromboxanes and leukotrienes, as we shall see. Here's another look at omega-3 fatty acids from the perspective of a senior who's been advised to eat healthy. Pickles is a comic strip about a couple of seniors by the names of Earl and Opal, and this is Opal on the right talking to her friend. She's just learned about omega-3 fatty acids and she thinks they should be renamed. So why are omega-3 and omega-6 fatty acids named as they are? Take a look at the ends of the chains of these molecules. This is linoleic acid, and this is carbon number 18 at the end of the chain. So this is 18, 17, 16, 15, 14, 13. But if you number at the end of the chain and count 1, 2, 3, 4, 5, 6, then the first double bond occurs at the 6th position from the end of the chain. Omega is the end, and so an omega-6 fatty acid has a double bond at the 6th position counting from the back of the chain. The same thing is true for arachidonic acid, as shown here. Omega-3s, of course, have their first double bond at the 3rd position from the end of the chain, thus the omega-3 nomenclature for alpha-linolenic acid, eicosapentaenoic acid, and docosahexaenoic acid, the latter two known commonly as EPA and DHA.